Alrighty. So, alrighty. This is my presentation on muscle histology. So the target objectives for this presentation, there are 4 of them. The first is to identify the cellular structures of skeletal muscle cells and describe the function of each structure. The second target objective is to draw a sarcomere and label all major proteins and regions of the sarcomere. The third objective is to relate the cellular structure of skeletal muscle cells, including the sarcomere, to the sliding filament theory. The fourth target objective is to describe the changes that occur to a sarcomere during contraction versus relaxation. There are a couple of vocabulary words for today's presentation. There is sarcolemma, sarcoplasm, sarcoplasmic reticulum, sarcomere, myofibril, T-tubules, triad, myofilament, myosin, actin, troponin, and tropomyosin. There are 3 types of muscle tissue. That's skeletal, smooth, and cardiac. And skeletal muscle, smooth, and cardiac are all made of fibers of tissue. The skeletal muscle is striated, meaning it's striped. It's tubular, and it's multinucleated. Multinucleated means that there are multiple nucleuses per cell of skeletal muscle. Skeletal muscle is voluntary and is usually attached to bone. Smooth muscle is non-striated and it's spindle-shaped, but it's uninucleated, meaning there's only one nucleus per smooth muscle cell. Smooth muscle is an involuntary muscle, and it is usually covering the walls of your internal organs, like your stomach, your liver, your intestines, your gallbladder, on and on, your esophagus, even. Cardiac muscle is striated, but it's branched, and it's uninucleated, meaning one nucleus per cell. It's an involuntary muscle, and it only covers the walls of a person's heart. So we're going to go over a skeletal muscle cell, and below there is pictured the macroscopic representation of a muscle cell's organization. So it starts from big and it gets smaller and smaller to the smallest unit, called a myofibril. I will start at the smallest unit and work towards the largest unit or unit of muscle or largest organization of muscle. So we'll start at the smallest, which is a muscle fiber. A muscle cell is often called a muscle fiber because it's long and cylindrical, as you can see in this picture beside me. On a muscle cell, the plasma membrane is called the sarcolemma, which is clearly labeled here in the yellow in the picture. The cytoplasm is called the sarcoplasm in a muscle fiber, and the smooth ER is called the sarcoplasmic reticulum, which on this picture is in blue. The little blue, it looks like veins on the side of the myofibril. The myofibril is a protein cable filling the sarcoplasm in the muscle cell. The arrangement of myofibrils is called a sarcomere. This is the larger organization of muscle cells. So we've gone over a muscle fiber, but muscle fibers are organized into a fascicle and then covered in endomysium. The endomysium is a layer of fascia around multiple muscle fibers. Then the fascicle is covered in a connective tissue called the paramecium. Multiple fascicles are then organized together into a bunch or in a group, and then wrapped in epimecium to form what we think of as a working muscle. The epimecium is a dense connective tissue covering it. The endomysium, the paramecium, and the epimecium in the end of the muscle will come together they will connect together, and then those will connect to the connective tissue in the tendon, and the tendon will connect to the periosteum, the outer layer of the bone it's being attached to. This is a picture of a myofibril. It is this is a picture of two myofibrils. Apologies. The T tubules are labeled in blue, and a T tubule travels down into the cell. It's filled with extracellular fluid, and that's to make sure that the sarcolemma can get close to the sarcoplasmic reticulum. Labeled in pink would be the sarcoplasmic reticulum. It's a specialized endoplasmic reticulum, or an ER, as referenced in normal cells, which stores, releases, and retrieves calcium ions. There's the triad labeled in yellow and blue. It is the arrangement of T tubules with sarcoplasmic reticulum membranes on either side of the T tubule. And then labeled in bright pink is a myofibril, showing there that I drew two, but a myofibril is just a singular cylindrical piece there. It contains a group of strand-like proteins, and each strand is made of repeating units called sarcomeres. This is a diagram of a sarcomere. So let's start with basic what they're made out of. So a sarcomere is made out of thin filaments and thick filaments, and Z discs or Z lines, as some textbooks reference. So the thick filament is labeled in blue, and it's made from strictly one protein called myosin. And as I blew it up here in the bottom, what the structure would look like, it's the trunk or the base of the thick filament has myosin heads on them, so they can attach to the thin filament here in black. The thin filament is made of three proteins: actin, troponin, tropomyosin, which I've blown up their structure also. The green little spherical proteins are the actin, and then all those little balls of actin are strung together using tropomyosin in purple, the purple lines, and then the troponin are little cylindrical proteins spread out all over the linking chains of the thin filament. The M line is labeled in purple, so that's the central part of a sarcomere. I'll talk about later about contraction in the M line. The H zone is labeled in blue, The two I bands are labeled in orange, and the A band is labeled in pink. So this is the sliding filament model. During contraction of the muscles, thin and thick filaments don't shorten, but they slide towards each other and overlap. The bands themselves, so these bands, the I band, the H band, will shorten during contraction, but filaments themselves don't shorten. So the thin filament and thick filament will only overlap. The filaments get closer to each other because myosin will bind to actin after the calcium ions expose the myosin binding sites. So we're going to talk about band lengths. During contraction, the Z discs get closer together. The H zone will shorten or possibly even disappear. So that would be this this piece here. We may all up and disappear, and these thin filaments may come the whole way to the middle, to the M line during contraction. The I band here and here, this would be the I band, will shorten during contraction, but the A band, this thick filament here, this in dark blue, will never change length ever. And there's my works cited. Thanks.